I can still be heard?
Yes, yes, continue.
Okay, Clostridium botulinum. We had covered Clostridium perfringens ovelchi and Clostridium tetani. So now we are covering Clostridium botulinum. And as an organism, it is motile because it has flagella. We said most of the species are flagellated and motile. And it also forms spores like the other species. And the position of the spore is either at the center or close to the end subterminal. There are the physical properties which are important. It is an organism that remains viable in contaminated parts of the environment for a long time because of those spores. And that contaminated part of the environment constitutes a significant source of contamination of various items. It has been associated with contaminated vegetables, contaminated foods, etc. And that contamination comes from that organism that is in the environment where those food items are placed. And these spores can withstand moist heat at 100 degrees centigrade for several hours. So they are relatively hardy spores, and they are destroyed at 120 degrees centigrade in 5 minutes. So it has antigens like the other species, identified and designated capital alphabetical letters A up to G. And those antigens are important when it comes to serotyping. And serotyping is important because of the toxins produced by each serotype. So these toxins are antigenically specific. So when you have a toxin produced by serotype A, it can only be neutralized by an antibody formed against it. An antibody formed against another serotype cannot neutralize a toxin formed by a different serotype. Then the exotoxin, it is the main virulence property, and it is referred to as botulinum toxin. It is also a neurotoxin, but it acts in a different way from that Clostridium tetani. It is a toxin which is heat labile, easily destroyed by heat at 100 degrees centigrade applied for 30 to 40 minutes. And the significance of this is that if this toxin is in the food and the food is properly cooked, heated hot, to a temperature that is higher than 80 for long enough, the toxin will be inactivated and the food will not cause any problem. So the clinical significance of Clostridium botulinum, the illness associated with it is called botulism. And this botulism is associated with food. So when it is seen, it's not a common illness, but when it is seen, most of the cases are related to food consumption of contaminated food. So that one is referred to as foodborne botulism. And it's a severe form of food poisoning, which develops when Clostridium botulinum spores, one, contaminate foods, which may be improperly cooked, improperly prepared before canning, and improperly preserved. It also develops when these spores germinate into bacilli. After contamination, when the conditions change and become favorable for germination, then the spores germinate into bacilli. And once they develop into bacilli, they begin to multiply in the food. And exotoxins are released by live multiplying organisms. And as they multiply, they release the botulinum toxin in the food. And then when the food is consumed, if it is not properly cooked, etc., one would consume the food containing botulinum toxin. and one will end up with manifestations due to the effects of this toxin. So this is a kind of foodborne illness or food poisoning that we described as one which results from preformed toxin. In other words, it's related to ingestion of the toxin that is already formed, unlike the others, which are due to toxins formed in the intestine. There's another type, which is relatively uncommon in comparison to the foodborne type, and this is wound botulism. And this occurs secondary to wound infection, like Clostridium botulinum. Like any other bacterium, it can infect a wound and colonize it and multiply there. And when it infects a wound, it lets the spores get into the wound and germinate there and produce bacilli, which release the exotoxin. And this exotoxin, whether it is foodborne or from a wound, it gets into the blood circulatory system and eventually gets to the nervous system. And its main site of action is presynaptic nerve endings of the peripheral nervous system and cranial nerves. And its main activity is inhibition of the release of the neurotransmitter acetylcholine. So any stimulation does not reach the muscle that it is supposed to reach, and as a result, the muscle is not stimulated and it becomes paralyzed. It's a different type of paralysis. From that of Clostridium tetani, it is due to overstimulation. So clinical manifestations will depend on the muscles which are affected and their impaired functions. So if muscles have functions and if they're paralyzed, then the functions will not occur, and that would be the manifestation. So it depends on the muscles. If it's muscles of the eye, there is interference with normal vision. If it's muscles that are involved in swallowing will become a problem, etc. So there's another type of botulism, but it's food related and it's called infant botulism. But as you read about foodborne botulism, you can also cover that one which affects some infants. So for lab investigation, where it is suspected as a type of foodborne or food poisoning, sometimes it involves a large number of people who have eaten a common dish. So there might be many people presenting with the same manifestations, and one needs to do a laboratory investigation. Stool is one type of specimen that can be examined. Other intestinal contents were available and also suspected food. And then the procedures will be the same, gram stain and microscopy. As for other clostridial infections, we have mentioned, they might be very scanty, but it is one method. Then culture is done again, as we have talked about. And then where there are specialized techniques, where facilities are better, they can detect the botulinum toxin. This can be done in the blood, it could be leftover food, or stool, diarrheal stool, or any other stool that would produce, because the toxin is formed in the food and it's ingested, it could be contained in the stool. Then some aspects of management, which is managed by giving antibodies to the toxin. And where the serotype is known, it should be a specific antitoxin to that particular serotype. Where it is not known, there are preparations which contain the antitoxin for all the serotypes. So the right one might take effect. For the wound botulism, one still does number one, administration of antitoxin, the specific causative serotype, and the addition, the wound is managed just the way other wounds are managed. Would require surgical intervention, then that will be done. And also antimicrobial agents to clear the Clostridium botulinum. Penicillin is effective as well as intranasal. Then the aspects of prevention, this mostly is directed towards the foodborne botulism. It is important that proper processing and canning of food are carried out. So it should be processed. If it is cooked food or the packaged when cooked, then it should be done under clean environment and bearing in mind that it could be contaminated, and if so, one should treat it in such a way that the contaminants are destroyed. And also canning of food. Sometimes it's associated with canned food, and that canning should be done, applying the necessary precautions under suitable environment. Then sufficient heating of food before consumption. We have just said that the toxin is susceptible to heat. So if there's food that is suspected to be contaminated, then it should be cooked properly to enable the toxin to be damaged. Then proper use of food preservatives in food factories. They should be used in effective quantities, but safe in concentration, safe in human use. Another species is Clostridium difficile. Could have a new name, but we'll just talk about it as Clostridium difficile. It is an organism that is part of the normal flora of the large bird, where the main other organisms is normal flora. But what is outstanding about it is that it's more resistant to commonly used antibiotics than other bacterial flora in the large intestine. So in a situation where broad spectrum antibiotics are administered for treatment of infection, most often it's in other parts that might get infected. Once it's administered, it goes throughout the body, even into the large gut. So there, it will inhibit the susceptible large intestinal bacterial flora. But because Clostridium difficile is more resistant, it remains uninhibited. And when now the others are inhibited, stress is created, and Clostridium difficile multiplies, and it is a proportion that now becomes significant. Each product reach a quantity that can now be effective, and it produces an enterotoxin and a cytotoxin. And these toxins act on large intestinal tract lining and exert their activity. As a result, there are many types of illnesses described as antibiotic-associated diarrheal illnesses. There are given many names in infants and others, but one of them is called pseudomembranous colitis. So the pseudomembranous colitis, that those toxins exert their activity on the lining of the large intestine and cause necrosis and diarrhea and all that is passed out, so the mucosal lining peels off together with the diarrheal stool, and it looks like a membrane on the skin. That is how it brought the name pseudomembranous colitis. So there's a treatment in terms of antibiotics, although it is due to broad spectrum antibiotics. There are also some antibiotics that are the ones that are causing it can be stopped for a while, and the effective ones. There are some which are effective against Clostridium difficile. Those can be used to reduce that Clostridium difficile or to end the diarrheal illness. Okay, the next group now is finished with the Clostridium or Clostridium species. They are gram-negative spore-forming anaerobic bacilli. Non-spore-forming, sorry, gram-negative, non-spore-forming anaerobic bacilli. Gram-negative bacilli do not form spores, so they can only be non-spore-forming. Species, there are many organisms in this group, and species of several genera encountered. They are encountered as normal flora in humans, together with other anaerobes in specific pods, and they are also encountered as pathogens. So these are good examples of organisms which are normal flora most of the time, but for one reason or another, they turn into pathogenic organisms. So there are a number of genera. Among them, we have Bacteroides, there's Porphyromonas, and there's Privotella. And remember, when you are classifying, we gave other examples like Fusobacterium. So there are quite a number. But these three here were one genus, Bacteroides. But there was reclassification based on differences in sub-biological and biochemical characteristics. Otherwise, under the microscope, they look similar. Morphologically, one cannot differentiate them. One has to apply those tests that will detect the biological and biochemical characteristics that are different among them. So if we look at Bacteroides, the species are non-motile, but they have a capsulated. And there are several species under Bacteroides still, despite the fact that the genus was divided. Still, there are many species under Bacteroides. One of them is Bacteroides fragilis. So they are normal flora, but at the same time pathogenic, and they have virulence properties like a capsule. You know what the capsule does. They have villi, villi. How they act as virulence factors or properties, and they have enzymes. But still, they cause various conditions in association with precipitated factors. Most of the time, despite the virulence property, they still need a precipitating factor in order to cause disease. And those precipitating factors include trauma, and also trauma, which is associated with the large intestinal tract that will cause injury of the large intestine. Secondly, the presence of devitalized tissue and the impairment of blood supply that also creates an environment that is suitable for the multiplication and disease position. And most of the time, again, it could be associated with injury of some sort. Then where there is an infectious process already going on, that is a pre-existing infection, and the organisms causing it are facultative anaerobes and other strict anaerobes, so one type or the other in inner tissues. Then bacteroides can also get involved in that infectious process. So one ends up with a mixed infection. So the presence of those organisms is important, the precipitating factor for it. So some of the clinical implications of Bacteroides fragilis, it has been associated with various infections as part of a mixed infection or a single causative agent. It has been detected in specimens taken from intra-abdominal conditions like appendicitis, peritonitis, and abscesses, and others. Has also been detected in specimens from other infections and abscesses involving inner parts, including organs like the lungs and the brain. And it has also played a role in infections which involve female reproductive system, whether they are related to childbirth or just infection in a particular part of the body. And there, the Bacteroides fragilis can also be isolated from blood as a causative agent of bacteremia. So the way the investigation is done is similar to those of the other organisms we have discussed, the Clostridium, fringes, etc., and the methods that we discussed for anaerobes in general. So the details would be the colonial morphology, effects on this and that. But those are now details for identification, where one isolates the organism and wishes to identify it and thinks it's Bacteroides fragilis. Then there are methods to follow, which are already present in various laboratory manuals. Then Fusobacterium, this is another one, gram-negative, transforming anaerobic bacillus, which shows a lot of pleomorphism in shape. Could be fusiform in shape, it could be a typical rod, it has lots of variation. So that is described as pleomorphism. But it's non-motile, it's gram-negative, and has many species again. Two of them, Fusobacterium nucleatum and Fusobacterium necrophorum. Now its significance, it's formed part of the normal flora where the anaerobic organisms are found, especially in the mouth, the mucosal surface in the mouth. And apart from being normal flora, it's also a causative agent of the infections. From that position of normal flora, it can become a causative agent of an infectious process. And this again can be as a single organism or with other organisms in a mixed infection. Fusobacterium nucleatum is one of the causative agents of ulcerative lesions in the mouth. It's a condition that was common many years back, where we had there was Fusobacterium nucleatum and some spirochetes, some other anaerobes causing ulcerative lesions in association with malnutrition, etc. Otherwise, it's an organism that responds to antibiotics like penicillin, like metronidazole, and also clindamycin. Then lastly, the anaerobic cocci. Again, these are together with the other anaerobes as normal flora. The gram-positive anaerobic cocci, there are many genera. One of them is Streptococcus. These numerous genera are differentiated by biochemical tests and metabolic end products, and sometimes it's hard to differentiate them, so they just refer to as gram-positive anaerobic cocci. And occasionally, they are implicated in some imposition of some abscesses. again, most of the time together with other anaerobes, like gram-negative bacilli. Then gram-negative anaerobic cocci, Veronella genus. The species is Veronella pavula. It is part of the normal flora, again, together with the others, and it can be isolated from specimens in a mixed infection, but it has no definite pathogenic role. It could be there together with the others, but it's not, its pathogenic role is not significant. It doesn't play any causative role in those conditions. And ladies and gentlemen, here we are going to talk this lecture on anaerobic bacteria for today. Why are you not talking? So did you hear this last bit? Hello? Can you hear me?
Yes, madam.
Yes.
Oh, okay. That's what we had for the lecture on anaerobic bacteria. The two lectures, we have covered all of it. What is remaining now is the practical part.